Hello, myself Gautam from 316. Today my topic is thermodynamics of melting of the double helix. The thermodynamics of melting a DNA double helix revolves around the process where the two strands separate or melt as the hydrogen bonds between complementary bases are broken. This melting process is essential in biological processes like DNA replication and transcription. The thermodynamics of DNA melting point delves into the interplay between energy, entropy, and molecular interactions, focusing on how these properties affect the stability and separation of the double helix. Here's a closer look at the critical thermodynamic factors and how they influence the melting process. The molecular interactions in DNA stability. There are two types: hydrogen bonding and base stacking and electrostatic repulsion. Hydrogen bonding. The hydrogen bonds between complementary bases provide stability to the double helix, while each bond is relatively weak individually. The cumulative effect of many hydrogen bonds is substantial. Second one, base stacking. Besides hydrogen bonds, base stacking interactions contribute significantly to DNA stability. These are non-covalent hydrophobic interactions between adjacent bases along each strand, leading to stabilizing van der Waals forces. Electrostatic repulsion. The phosphate backbone of DNA carries a negative charge which creates repulsion processes between the strands. Quaternions, typically metal ions like magnesium, sodium, stabilizing the double helix by neutralizing some of the repulsion. Second is thermodynamics parameters and melting temperature. First one is enthalpy. The enthalpic component of DNA melting reflects the energy required to break hydrogen bonds and disrupt this base stacking interactions. Typically, higher the entropy value indicates more stable DNA with a higher temperature. Second one is entropy. The entropic component is driven by the increase in disorder as the structured double helix separates into two single strands. This increase in disorder favors the melting process. Third one is free energy. The Gibbs free energy of melting dictates whether the melting process is thermodynamically favorable at a given temperature. DNA is stable when free energy is negative at physiological temperatures, but as the temperature rises to approaches to zero and the system reaches equilibrium between double and single stranded forms. Nearest third one is nearest neighbor thermodynamic model. This model calculates the stability of the DNA duplex based on each pair's unique interactions with its neighbors. It takes into account both sequence specific interactions and the influence of adjacent bases on each other's melting behavior. Stacking free energies. Each base pair's stability is highly dependent on the pairs flanking it due to base stacking, making nearest neighbors calculations essential for accurate predictions. Fourth one is impact of external conditions on DNA melting. Salt concentration. Higher concentration of cations stabilize the DNA duplex by reducing the repulsive forces between negatively charged phosphate groups or the DNA backbone. pH. Extreme pH levels can destabilize DNA by affecting hydrogen bonding. For instance, highly acidic or basic conditions can protonate or deprotonate the bases, disrupting base pairing. Organic solvents. Certain solvents, example formamide, disrupt hydrogen bonds, lowering temperature by weakening the double helix. Fifth one is applications of DNA melting thermodynamics. There are three types: polymerase chain reaction, DNA sequencing, hybridization, evolution, and genome stability. First one, polymerase chain reaction. Knowing the DNA sequence is crucial for setting the appropriate annealing and denaturation temperatures in PCR to ensure specific and efficient amplification. DNA sequencing and hybridization. Melting thermodynamics inform probe design in techniques such as fluorescence in situ hybridization and microarray technology, where precise hybridization is essential for detecting specific sequences. Evolution and genome stability. GC content varies across species and genome regions, impacting the thermal stability of DNA and reflecting adaptation to environmental conditions. The study of DNA melting thermodynamics is not only fundamental to understanding DNA stability and function but also underpins various molecular biotechnological techniques and the design, diagnostics, and therapeutic strategies in medicine. My second topic is kinetics of unwinding of the double helix. The kinetics of unwinding the DNA helix is crucial in processes like replication, transcription, and recombination. DNA unwinding involves breaking the hydrogen bonds between complementary base pairs and disrupting the helical structure, requiring energy and specialized enzymes. Key factors and steps in DNA unwinding kinetics. First one is enzyme catalysis by helicase. Helicases are the primary enzymes responsible for unwinding DNA. They move along the DNA strand powered by ATP hydrolysis and break the hydrogen bonds between base pairs. The kinetics of unwinding is directly linked to the rate of ATP hydrolysis. Faster ATP hydrolysis generally corresponds to faster unwinding. Helicases can vary in processivity, which affects how long they remain attached to DNA without associating. Second one is influence of supercoiling. DNA supercoiling can either enhance or higher the unwinding process. Positive supercoils increases tension, which can resist unwinding. Topoisomers work alongside helicase by relaxing supercoils, allowing more efficient unwinding and preventing excessive strain on the DNA. Third one is binding proteins. Single-stranded DNA binding proteins stabilize the unwound single strands by preventing reannealing and protecting them from degradation. These proteins help maintain the unwound DNA state long enough for application for or transcription enzymes to process it. Fourth one is temperature and ionic conditions. Higher temperatures can increase the rate of unwinding as the double helix becomes more flexible, but temperatures too high may denature DNA. The presence of ions affects helicase activity and stability of the double helix, with optimized ionic conditions required for efficient unwinding. Fifth one is rate limiting steps. The initiation of unwinding, where helicase binds and destabilizes the helix, is often slower than the subsequent steps of continuous unwinding. Helicase pausing or dissociation, especially in regions with strong secondary structures, can slow down unwinding kinetics. Sixth one is kinetics model. Kinetic studies often model unwinding as a multi-step process involving binding, conformational changes, ATP hydrolysis, and transcription. These models provide insight to the rate constants and energetic barriers which influence the overall speed and efficiency of unwinding. Seventh one is regulation of DNA unwinding. Cells regulate unwinding at multiple levels. For instance, helicase loading at replication origins is carefully controlled to ensure that DNA is only replicated once per cell cycle. Post-translation modifications such as phosphorylation can regulate helicase activity. For example, phosphorylation of the MCM complex affects its helicase activity and helps coordinate unwinding with replication needs. Eighth one is challenges in DNA unwinding. Secondary structures. DNA sequences rich in GC pairs or containing repetitive motifs can form secondary structures, such as hairpins, that complicate unwinding. Lesions and damage. DNA damage, such as pyrimidine dimers from UV light or crosslinks, can create block that halts helicase. Specialized helicases, such as those involved in DNA repair, can bypass or remove these obstacles. Ninth one is biophysical studies on unwinding kinetics. Techniques like single molecule fluorescence and atomic force microscopy have allowed scientists to directly observe DNA unwinding in real time. Tenth one is clinical applications. Errors in DNA unwinding can lead to mutation, genomic instability, and cancer. Many diseases, including Bloom syndrome and Werner syndrome, result from mutations in gene encoding. At the end, studying DNA unwinding kinetics is essential for understanding the dynamics of replication and transcription machinery and how cells regulate this process under different physiological conditions. Thank you.